Today we're going to talk about DNA synthesis, or how our DNA is copied. So our first image here is a helix and then a section of that helix zoomed in so that we can see the bases. So the first step to replicating DNA is we have to break our original DNA, and that is done by an enzyme called helicase, which goes down the helix and it breaks these hydrogen bonds between these bases. And it's going to keep just going down the line. And it's important to note that while helicase is unwinding, there's another enzyme called DNA gyrase, which is easing the tension that's caused by unwinding each section. So that's an important part to remember. And so we get to this completely separated section of DNA, and now we have to start combining things. It is important to notice that DNA is directional. So we have 5' and 3' ends. So our DNA polymerase is going to add new bases so that we have new DNA strings, but it can only move from 5' to 3'. So it's going to go down the line, and it's going to add the complementary base to whatever base is open from our original DNA strand. Now, this bottom section of DNA is called our lagging strand because the DNA polymerase is only able to move so much with the unwinding, so it has to keep stopping and repriming and then moving again. So it leaves us with these fragments, whereas our top strand is just going to have one continuous motion of adding. So at the end, you have right here is our leading strand. When the DNA polymerase is done, we have the functional DNA, it's all there, and then we have our lagging strand, which is kind of in pieces. So what we have to do is take our friend DNA ligase, and it's essentially like the glue for the DNA. So it's going to go ahead and stick these fragments together. So it's going to travel down our lagging strand and seal these fragments, and then we're going to end up with, just like our leading strand, we will now have a continuous piece of DNA. And then the helicase will continue to move down the helix until it's all replicated.